Wow. Okay. Since I'm gonna have to announce it because I'm gonna end that question. So does everyone see that? 68% of us are taking alectinib, 20% are on lorlatinib, 6% are on bernactinib, and then basically that's about it. The next question: um, what's the length of your disease? If you pop in where you're at. All right, I'm going to end that poll. As you can see, the majority are 1 to 3 years, 38%. Then second is 1 year and under, it's 27%. And then we got 18 and 17%, basically 3 and 5+ years. So there's one more question. Oh, oops. Last question is, how many numbers of TKIs have you taken? If you've taken chemo plus a TKI during your treatment, please indicate, or immunotherapy. Okay, the numbers are changing. Okay, I'm going to end this poll. The majority of us at 62% have taken one TKI, and then 23% is two TKIs. And then also chemo is 26%, which is very high. Personally, like myself, I've taken 4 TKIs, so I'd be at the 5%. So that is kind of the polling of where we are. Right now, we have 121 members that joined our meeting here. And I've hit the recording button. Just want to let everyone know that this is being recorded. And if for some reason, you get unmuted and you start talking, you will be on the recording. I want to introduce Gina Hollenbach. She's our president of ALK Positive. You can just wave to us or say hello. Colin Barton is joining us. He's also our head of our medical department for ALK Positive. And then Amanda Nerstad will be monitoring us. Go ahead, Amanda, take it away.
All right. Hi. I think Gina was going to talk about our ALK Positive group real quick.
Yeah, my name's Gina Hollenbeck, you guys. It's so good to see everybody. I'm the president of ALK Positive Incorporated, which is a group of incredibly talented people from the support group who are working really, really hard to make sure that ALK Positive cancer is one day a curable disease. So we're hoping to improve the life expectancy and quality of life of ALK positive patients worldwide. And I'm wearing some awesome gear here in case you don't have any. You can go to alkpositive.org, and when you go there, you just hit shop, and we have an awesome shop there where you can grab some stuff, and that stuff is going to, all the proceeds from that are going to go to fund research, like the research that Dr. Lovely is going to talk about. And so it's so exciting that I get to be the president of this incredible group of people who really do work hard. And it gives me so much hope to know that, you know, we're working together to make sure that ALK positive is one day a curable disease. So that's what we're really working for. It's really great to see all of you. I see some of you, I see a lot of patients and caregivers, and it just, this is one of my favorite times because I can see all of us together. And I know that some of you guys are really healthy and doing great. And that gives me so much hope. It's so exciting. And I know that some of you are not doing so great. And I know some of you are going through some really scary times right now. And some of you are about to go through surgery this week. And the cool thing is that COVID has kind of brought us together, gotten us acquainted with Zoom so that we can do some of these things. And I know that together we're stronger. And so I'm so excited about all of the hope that we're going to get and that we're going to hear about tonight. So So Amanda, I'll give it over to you. It's good to see everybody.
Yay. Hi, my name is Amanda Nerstad. I'm also an ALK positive patient. I was diagnosed in 2016. And I agree with Gina. It is so nice to see faces. We're always within our group, so it's nice to see everyone's faces. And I'm so excited to introduce Dr. Christine Lovely. She's an amazing oncologist and researcher at Vanderbilt Ingram Cancer Center in Nashville, Tennessee. She has a brilliant mind, but also a heart of gold. For those of you, if you're able to, she has opened up her lovely lab for the past 2 years for ALK patients. And if you're able to, please do it. It's an amazing opportunity to see ALK research being done. And when you go into her office, she has this big board, and it's, you know, and you just assume that it's her family photos of, you know, family members, but it's all of her patients. And she says, you know, I do this as a reminder. This is why I do what I do, which is amazing. She has a heart of gold. So she's going to talk a lot about herself and her research. And also we're excited for her to talk about the research that she and Dr. Bavona are doing that our ALK Positive group funded. So Dr. Lovely, it's all yours.
Everyone. So Amanda and Gina, thank you so much for that really kind introduction. I'm already like tearing up. So it is really such a pleasure to be here. Thank you all so much for sharing your Sunday night with us when you could be with your family or watching football or both. I hope this will turn out to be a really valuable time spent for you. It is so nice to see everyone's face too. A lot of times we do these Zoom meetings and I can see me, but I can't see everybody else. So I love seeing all your beautiful faces, and please pipe in and ask questions. This is meant to be totally informal. I'm at home, I'm in my sweats, and my dog is in the background. And so this is not like a formal seminar or office visit. It is really just meant to be a discussion amongst friends. And I am absolutely delighted to be here tonight. And thank you all so much for being part of this. I have a little bit of a presentation to show. I don't want to take up too much time going through slides because I want this to be more interactive. But to Amanda's point, my lab, we love having people visit. I think everybody's been to a doctor office, whether you like to be or not. Everyone knows what it's like to go to the doctor and to be in a hospital, to be in a clinic, but very few people have actually been to a research lab. And so I think it's really important that we share and open up the labs to see, what does that mean? What does research look like in implementation? You know, who's doing the research? What are they actually doing? And what do their days look like? And we loved having a big ALK positive group in February 2020 come and visit. And we had like a 4 or 5 hour tour of different parts of the research world at Vanderbilt. I hope that in the post-COVID days, we can do that again, because it was not only fun for me and fun for all the ALK positive patients and caregivers who are there, but it's so important for laboratory people who are not also in the clinic to see patients that are alive and doing well and thriving. It's very, very motivating for my trainees to also see, wow, I mean, after Amanda and Gina were there, they were like, I can't believe that they're even lung cancer patients. And that's very inspirational for them, very important for me to make sure that they see both sides of this equation. Why are we doing the work that we do, and why do we need to work harder and harder on it, so that we can really help our friends and colleagues and community to battle this disease. So I'm going to share my screen and start with a little bit of a virtual lab tour. Again, don't want to take up too much time, maybe 15 minutes maximum, so we can have at least a half an hour for questions and answers. That's Tegan, my dog. Okay. Can everyone see my screen now?
Yes.
Okay, so let me move around my toolbars. So this is going to be a virtual tour of the lab. It is not anywhere near as good as being there in person, but just thought it would be cool to show you, what does a lab look like, and what does a lab that studies ALK look like. So first and foremost is who's in the lab and who actually does this work. And in most research labs, my own lab included, we have people who are getting their PhDs, so students who are working to study lung cancer to get their PhD. We have people who are what we call postdocs. So they have gotten a PhD in research science, but now they're wanting to do some more advanced training. And then there are levels beyond that. Like when you're finished being a postdoc, you become an instructor or a staff scientist. And so just to introduce you to some of the people in my lab and show you kind of the, who's in there and what are they doing. These are people that I supervise, and they supervise me sometimes. They are building the research and bringing the enthusiasm. So Juan is the primary person who works on ALK in my lab right now. He's a very experienced scientist who's been doing science for the better part of 2 decades. Yoon Kai is next. He works on ALK a little bit and helps with some of the studies with Dr. Bavona's lab, but he is predominantly doing work on EGFR, but thinking a lot about drug-tolerant persister cells in any type of lung cancer. And I put here what the hobbies are, because I think it's good to see people both as their professional side and then what they really enjoy doing in their life as well. Henry, if you're on Twitter, Henry's a really big voice on Twitter, started something called Black in Cancer. He's the newest postdoc to my lab, came from Tuskegee. He's thinking about combination therapies. Let's see, who else do we have? Zhen Fong, another postdoc. He studies a lot of protein structure function with respect to EGFR, some ALK, some ROS1. Prasad studies small cell. We'll show you a little bit of everybody's work, very high level. Yang Jin is the heart and soul of the lab. She is the research assistant who kind of coordinates the day-to-day lab activities, who helped me put together these slides. Portia is an MD PhD student, so she is like myself, doing both medical school and graduate school. She studies small cells well, and it's just been such a joy to work with. People we work with a ton. So Wade Iams he has been my mentee for almost 10 years now. He's now on faculty. He's a thoracic oncologist at Vanderbilt as well. Keep your ears on him. He's a superstar. Josh Bauer is a colleague and friend who helps us with doing some of the drug screens I'm going to show you about. So he runs what we call a core lab at Vanderbilt, which is not an individual investigator, but a lab that services all the investigators. And there's about 350 different laboratories at Vanderbilt. And then Dave, who's this guy here with the beard, he was in my lab, was a postdoc, and then took a job in the core and really has been hugely helpful for all the ALK studies that I'm about to show you. Okay, so we grow tumor cells in the lab, and we grow cells that are from patients. And this is just a picture of where do the cells live and how do we grow them. So on the left-hand slide here, this is what we call an incubator. It is body temperature, and these little round things inside are plates where the cells are growing. They like to have a certain level of oxygen, a certain level of carbon dioxide, a certain temperature, and these machines can regulate all of that. And so this is where we grow the cells and passage them, and they can grow indefinitely. They just keep growing and growing, which is both good and bad. But these are the cells that we use for our experiments, and I'm going to show you some ALK cells in a moment. We handle the cells in something called the biosafety cabinet. So this is a sterile environment, so we don't want to contaminate the tumor cells that we get from patients. They're so precious. We don't want to contaminate them with stuff that's on our body or bacteria that's on our skin. And so we handle the tumor cells in these biosafety cabinets that provide a sterile environment and allow us to work with the cells very carefully. And so in our world, cells are like gold because we need something to be able to study the tumor. And tumor is gold, but sometimes we actually get cells from the tumor that we can grow and grow and grow in the lab. And this is how we handle them. Here is an actual picture of ALK positive lung cancer cells. So these are some of the cells that we use for the experiments I'm going to show you. And so in this case, we have made the cells grow. These are just normal growing cells in the incubator that I just showed you. We added tags to the cells so that we could look at them on individual levels. So if you look where my pointer is here, there's like a red circle in the middle of the cells. That's a label in the cells, and that allows us to follow the cells on a cell-by-cell basis. So instead of looking at a whole population of cells, instead of looking at 1,000 cells at one time, we can pick one cell and follow whatever that cell is doing when we treat it with drug. And these three are all from patients. So this STE1 was from one of the first ALK positive patients I ever treated. STM is from another ALK positive patient, and cute story, these cells, and she's very vocal about this, so I'm not breaking any confidentiality. These STM cells were made from a patient of mine and his wife and their 2 kids. The kids were very young when he passed away. It was in the early days of ALK. And his wife and the 2 kids, they live in Louisville, Kentucky, which is about 3.5 hours from Nashville. They come twice a year to visit the cells. And they have been doing that since he passed away, which was almost 10 years ago now.
Wow.
And when COVID finishes, this is such a great story. When COVID is over, his oldest daughter is going to come and work in the lab. And so that will be so wonderful to, have her experience, like actually taking care of her dad's cells and being part of the research and just seeing how, how do we get more really bright, really enthusiastic minds into this, because it takes a village and we need a lot of people to solve these problems. So these are some actual pictures of ALK positive cells. Here is an example of a tumor. So here's another thing I think a lot of people have not seen, but this is actually on the left-hand side. This is what your tumor looks like when you have a tumor biopsy. So imagine you're getting, a biopsy in radiology and they're doing it CT guided, or you're getting an ultrasound biopsy. They take a piece of the tumor, a core, and they put it in something called paraffin, which is like glue. That's the kind of white glue stuff around here. And then in the middle of that glue is the tumor. So these are 4 different tumors. You can see they all look very, very different. So here's one tumor, it's kind of diffuse. It's taking up the whole block. Here's one tumor, it looks like a rectangle. Here's one tumor that's like a long squiggly line. And this one here is actually several different dots of a tumor. So they basically took multiple biopsies and then put it onto this slide. So this is what everybody who had a biopsy, this is what your biopsy actually looks like. When we take it out and it's stored in pathology in something we call FFPE, or formalin fixed paraffin embedded. When you hear FFPE, this is what you're talking about. It's just these little blocks of tumor cassettes. And here's what we do with them, or one of the things we can do with it. So we can actually take a slice of the tumor and basically stain it for different markers. So in this case, we took a slice of the tumor and we're asking, can we look at ALK within this tumor? And so if you see here on the bottom, it says ALK IHC 20X. What that means is this is a tumor that's been stained for ALK. IHC stands for immunohistochemistry. That's just the technique that we use. And 20X is the magnification under the microscope. And so this is what your ALK positive tumor looks like when we look at it under the microscope and when we store it after you've had the biopsy. A couple of other just techniques. I think it's helpful to know, what are they doing in the lab? We look at DNA, RNA, and protein. Those are the building blocks of every tumor and every cell in your body. And one way we look at DNA is by something called an agarose gel. So each of these bands, these little white bands here, are pieces of DNA that we separate by size. That's a very standard technique in the lab. And then here, without getting into too much detail, this is what we call a Western blot. So if we want to take those cells that I showed you that were in culture, and we want to actually look at the proteins inside the cells, one other way we can do that is by basically busting open the cells and looking at every protein that's inside of them. And this is an example where we bust open the cells in this gel, what we call a gel up here. So this is the actual gel on the top right. Bust open the cells, and I'll be happy to go through this in more detail, but basically what we're looking at in these black and white films is just ALK protein in the cells. And this is a very, very standard technique in the laboratory. One of the many ways we can look at ALK. One more thing, and then I'm going to get into a little bit more detail about some of the ALK questions we ask. So another thing we do in my lab is we look at circulating tumor cells. This is an example with small cell lung cancer, but you could look at this in other tumor types as well. And the reason we do this with small cell now is because small cell tends to have more circulating tumor cells than non-small cell in general. But this is a machine called the Rare Site, where we actually take blood samples from patients and we look at circulating tumor cells in the blood. So we actually can take those cells, and again, I'll be happy to go into a lot of detail if you're interested, but we take the cells and we look at different markers on the cells to say, is it a tumor cell in the blood? And if it is, what kind of tumor cell in the blood is it, basically? So just a couple high level, this is techniques that we use in the lab. This is what a lab looks like, and these are the people who are in it. We could please come and see us in person because it's so much more impactful when you actually see what a research lab looks like. But I think it's helpful to at least start to know, like, well, what are they actually doing there, and what techniques do they use? Because most people have not had the opportunity to ever see a research lab before or know who's there and what they're doing. Okay, so part of our research, and all researchers love to talk about their research. And so you actually have to constrain people and say, cut back, rather than like get them to talk about it. And so I'm going to show you a few slides. It's going to be high level overview because we could talk for a full hour just about the research, but I wanted to frame the research around who's doing it and what are the techniques we use to really, help you to understand, like, if you're going to fund the research, you should know what people are doing in the research lab. And so this is part of our funded research project working with Dr. Bavona. This is something that I showed you a picture of Josh and Dave, my colleagues, a few slides back. This is something called a high throughput drug screen. And so my lab, working with this core lab, we can do an experiment where we say, let's take the ALK cells and let's test them with thousands of drugs at the same time and see what drugs kill the cells. It's really that, it sounds so simple when you say it, but that's really what we're doing. It's a lot more detailed than that because we have to prep the cells in certain ways. And I showed you the cells that like had the red marker. Those are the cells we use for this type of experiment because we want to be able to look at individual cells one by one. And so we take these plates that can hold 384 different, they're like little itty bitty wells, your eyes can barely see them. And we plate the cells and we put in compounds, drugs. Most of them have already been tested. They're FDA approved or they've been in clinical trial. And we're asking, very broadly speaking, broad strokes, is how do we kill ALK positive cells? In addition to ALK inhibitors, that's a given, but how do we actually, think about what is going to kill these cells? And do that in a very high throughput method. And high throughput is a word we use a lot in science. It basically means instead of doing it, something 10 times, do it 10,000 times or 10 million times. We want to be very broad and as fast as possible. For human hands to do some of these experiments, you couldn't do it because no human can plate 10 million samples at the same time, but a machine can. And this is all robotics that we use. And this was part of the tour we actually had when everyone came last year. So it's a high throughput drug screen using robotic technology to look at individual cells to ask what kills ALK positive cells. And so that's high level, and then we distill that down. Well, what actually worked in our initial studies? We get a list of drugs and they're ranked by what worked the best, and what worked the best is what killed the most versus what didn't work at all, what didn't even make the cells not grow. And so this is a table. It's not meant to be very granular. It's just meant to show you some of the output that we get, where we, if we're doing 100,000 compounds at the same time, we have to be able to look at that data in a very detailed way. And so we rank them about what drugs work the best, really as simple as that. And one of the classes of drugs that worked really well in this drug screen was something called aurora kinase inhibitors. Like Aurora, like I think that's the name of Cinderella or Snow White, one of the Disney princesses. It's also the name of a protein that works in the cell cycle, and it's a protein that actually is important. When your one cell becomes two, it goes through a process called mitosis, where you have one tumor cell and it just splits and it divides and it becomes the mother cell and the daughter cell. But when you're talking about mitosis, this protein, aurora kinase, is a very important protein for mitosis. And it was one of the biggest hits, and by hit, I mean, it was one of the best compounds that had activity against the ALK positive cells in our drug screen. And so then that's great. So we have a hit, we have a potential target. What do we do next? Well, we go back to the cells. And so this is an experiment, and I'll walk through this a little bit slowly, but wanted to actually show you, like, what does the data look like that we get? Because I think it's also important to know when you're thinking about research and when you're evaluating research, when you're serving on like a study section, what are people showing you and how do you interpret that data at all? What are the strengths and weaknesses? And so this is H3122 cells, which are ALK positive cells, seeded at 2 × 10⁵. So that's just the number of cells in the plate. And they were treated with different drugs. And so if we look at this graph, sorry, I'm trying to move around on the bottom here, the x-axis, if you think back to basic mathematics. The x-axis is different drugs, and the y-axis is the cell number. So let's look at this for the left part for a second. DMSO, that's just the control. So that's not a drug. That's what we call the solvent. It's what the drugs are dissolved in. And then we have alectinib at a concentration 500. We have lorlatinib at a concentration 300. We have brigatinib at a concentration 300. And then borisertib, which is one of the aurora kinase inhibitors that came out of our drug screen. And what you can see here is compared to this bar, which is the control, so these are just normal cells, all of these, the ALK TKIs and borisertib, all kill the ALK cells. Well, that's great, but putting two drugs together is kind of hard. As you all know, there's some great questions about combination therapies. Two drugs together can be very toxic. Two drugs together can cause more side effects than one drug. And so something that's very important to us is how can we get the best effect at the lowest dose? And so we're thinking about not just what kills the cells, but how do we maximize the amount of cell death, but minimize the amount of drug that we need to deliver to get that effect? Because we all know when you put two things together, whatever, you're talking about Tylenol and aspirin or Benadryl and Tylenol, you're going to get more effect than one drug alone when you put two together. That's one of the major difficulties with combination drug trials. That's one of the major reasons they move so slowly. So here, if we look and we're thinking about, can we alter the doses? Here, if you see on the bottom again, alectinib at 15 compared to 500, lorlatinib at 3, brigatinib at 4, borisertib at 60. And so compared to the control over here, all four of these concentrations, when we use only one drug, basically has no effect. But look what happens when we put the low concentrations together. So for example, this is alectinib 15 with borisertib 60. Look at how much it kills the cells. Whereas we needed 500 of alectinib alone to get that effect over here. And I hope that makes sense. I'm going a little bit fast, and I'm from New York, so I tend to talk fast. But the idea is find drugs that kill the cells and then make sure that we're thinking and being very thoughtful about what is the dose and how do we maximize the effect while minimizing the dose. So one other thing we're doing as part of our project is genomic studies, but not the genomic studies you think about for the clinic. In the clinic, we predominantly think about DNA changes. And that's what we can measure more easily with the FFPE tumor blocks that I showed you earlier. In the lab, we can look at other things, other analytes, we call them, not just DNA. We can look at something called RNA. And RNA is really important because RNA says how much of a given, goes DNA, RNA, protein. RNA says how much of a given target like ALK is present in the cells or not. And so you have to have all three of those factors together. And let me give you an example. You can have a mutation, a DNA mutation in a cell that's not expressed. So the DNA mutation will be there, but the RNA will be almost zero. So the fact that the mutation is there doesn't really matter because there's no RNA, which ultimately goes to the protein. And so we wanted to understand what's happening at the RNA level and how can that help us to understand drug resistance? Because I would argue quite strongly actually is that looking at DNA alone has brought us somewhere, like all the ALK mutations that confer resistance we find by DNA, but it hasn't been a home run. And we need other ways to think about what's causing resistance other than just mutations in DNA. Even in other tumor types like EGFR, just looking at DNA has not been a home run, right? We have P790M and MET amplification, but that's a small percentage of patients with resistance. And so we feel like we need to paint broader strokes. What else can we look at that may be mediating resistance? And changes in RNA levels could be one of those. And what this is showing you here, this complex slide, is what we call a heat map. You see these all the time in science. And what the red is, red boxes are genes that are upregulated. Blue boxes are genes that are downregulated. And so if we ask, I have an ALK positive cell line that's sensitive to ALK TKI, and I have an ALK positive cell line that's resistant to ALK TKI, what are the differences between those two when it comes to the RNA expression? And so all of the genes here are genes that are upregulated in the ALK TKI resistant cells, meaning they're higher in the resistant cells than in the TKI sensitive cells. And the blue is all those genes that are downregulated, so they're lower in the resistant cells than they are in the sensitive cells. And this is one way we use this and something we look at all the time in research. And to to go a little bit more detailed, along this column, the y-axis here is all different genes that are up or down regulated, and the x-axis, the bottom, is different cell lines. And if you hear some like growling in the background, I'm sorry, that's Tegan. We're going to give him a treat, so he'll be quiet. Sorry about that. But this is something called the heat map, very, very commonly used in science. So what did we do with that heat map? And I'm going to go back to my Western blot. And so this is a method we use to look at protein. And one of the genes that came out of this RNA study from our heat map was something called clusterin. It is a gene that protects cells from undergoing programmed cell death or apoptosis. So clusterin basically protects the cells. When it's present, it can help the cells not die. Well, that's bad because we want cells to die. So we don't want cells to change something that's going to protect themselves. We want to get rid of the protection. We want those tumor cells to die. But tumors are very simple. Tumors want to live. That's all they want to do. They want to do all the nasty things that tumors do. They grow, they live, they spread. They're going to find a way to do it, and we have to find a way to prevent them from doing that. And so they're going to change things in the tumor that's going to cause the tumor to have the ability to not die. And clusterin is an example of one of those. And so here you can see this is an ALK positive cell line treated with all 6 different ALK TKIs. So crizotinib, alectinib, ceritinib, brigatinib, lorlatinib, and sortinib. And let's just look at brigatinib down here in the bottom right for a moment. What we see here is this is time. So H3122 is the ALK positive cell line. And then here's hours. So 0 hours, 2 hours, 4 hours, 8 hours, 24 hours, 48 hours, 72 hours of the drug treatment. This top bar here is ALK. And so ALK goes away. The phosphorylation, which is a measure of ALK activity, goes away with the drug. That's totally what we would expect, right? What we see here with clusterin is it starts at basically nothing. And then by 72 hours, you can see the difference. It's a huge difference between how much clusterin is there at 0 and how much clusterin is there at 72. Well, this is a gene that gets upregulated to protect cells from dying. So that's not what we want. We don't want to protect the cells. We want to take away their protection mechanisms. So how do we do that in the lab? Well, one way we can do that is by something called gene knockdown. And this is a pretty cool technique we use in my lab that just looks at fluorescently labeled cells. I'm going to show you this in a different way, but basically what I want you to, the take home message is that when we knock down clusterin, the cells die more, and more dead cells on this type of graph is shown here in yellow. And so when we treat the cells with ALK TKI and get rid of clusterin, so take away the protection, the cells die more. Let me show you that in a different way. So here is my same cells again, H3122. They're kind of our ALK positive cell line workhorse. Here's clusterin, and shown here in this black line. And here's what we call knockdown, or SH clusterin, which basically means we take the gene away altogether. What happens to the growth of these cells when we take the gene away altogether? Remember, clusterin protects cells from dying. So we don't want cells to live. We want to take away the protection and make the cells die more. So let's see what happens. So here's a graph, and along the x-axis is different treatments, and the y-axis is the cell number. So the first is just alectinib treatment. So if we treat the ALK positive cells with alectinib, here's the number of cells we get. If we treat the cells with alectinib and we do something called SH clusterin, or knock down clusterin, take away the protection factor, the cells die more, which is totally what we would have predicted. So cells, the tumor cells, ALK positive tumor cells, they upregulate this gene called clusterin to help protect the cells from dying. And if we remove the ability of the ALK cells to upregulate that gene, the cells die more. So this bar, this big bar here, the tall bar, is alectinib only. The 2 other bars are alectinib, same dose, when we remove the gene clusterin from those cells. So we remove the protection factor. I think that is my last data slide. So here's just my, hopefully that's within the 15-minute time frame. Here is my research is fun slide. So this is various pictures in my lab over time. It really takes a village to do any of this work. I've been blessed to have really fun, smart people to work with at Vanderbilt and across the country and across the world. And I think one of my favorite things about the lung cancer community is just how collaborative everyone is, which is fantastic because we are all here to help our patients. And that's ultimately means we have to share and work together and do everything we can as a team to make this happen. And here's a picture from our visit last year. So you'll see some friendly, familiar faces. We just love having everyone and hope that in the post-COVID days, everyone's able to visit again. And I think that will be my last slide. Yes.
Well, thank you very much. That was very impressive. Very impressive. Thank you.
Over time. I'm so sorry.
It was great. No, so we asked people in our ALK group to comment in our comment section some questions. So we're going to try to get through that. And we also want to be respectful of your time. So you basically answered the first question from Craig Smith, Craig in Canada. But just as a follow-up, is preliminary data available to indicate outcomes with any of the ALK-TKI combination research and clinical trials? Which combos do you feel are the most promising? Is information building to change the standards of care within the ALK community based on this research?
Yeah, this is Craig. Thank you. This is a great question. It's a loaded question. I will say we could spend an hour talking about this question alone. Number one, combination trials tend to run more slowly because every time we put two drugs together, we have to go back to the beginning. And so every drug goes through a phase 1. Every combination has to go through a phase 1. And so you can't start where you stopped off, if you will. You have to sort of go back a few steps. And that can often cause some delays because it means the trials have to get up and going, patients have to be accrued, we have to see if it's safe, and it's all around safety because like I said, when you put two drugs together, you will get more toxicity than one drug alone. Right now, what I think are the currently in trials, the things I'm excited about is ALK plus MEK inhibitors. And the MEK inhibitors are trametinib and binimetinib. There's another one called selumetinib. I don't like that drug. It tends to have more toxicity for biochemical reasons. The issue with ALK plus MEK is we use that actually combination in other tumor types. So it's a standard of care in a type of melanoma that's an oncogene-driven melanoma. So we have a lot of information about how to safely deliver MEK inhibitors. There's a lot of preclinical data, laboratory data from several labs, including my own, that ALK inhibitors plus MEK inhibitors work better together, and the trials are underway. So I'm excited to see how those go. And they're both in the first line setting, meaning the patient who's never seen any ALK-TKI before, but then also at the time of resistance. And I think it will, fingers crossed, work in both settings if the toxicity is okay. The other combination, which is just moving into the ALK world now, is a totally new drug called a SHP2 inhibitor. It is a drug that's predominantly right now, the one that we know the most about is made by Novartis, which, as you know, is located in Boston. So that a lot of the Novartis studies will go right to MGH, which is awesome because then we can just move them from the drug company to our wonderful colleagues there. The ALK plus SHP2 inhibitors is another one I'm excited to see move forward. I think, we would all love to see even more and more, but honestly, and this is Christine editorializing a little bit, we need a unified front across this, right? Having a combination trial at one institution is fantastic. Having a combination trial that is open across the United States is even better because it makes us get the information better, faster, help more patients. And so these trials that are currently present, the ALK-MEK and ALK-SHP2, they're at single institutions or maybe one or two institutions. I personally would love to see, for example, like the NCI ALK Master Protocol have combinations because it's open at hundreds of sites. Just give more patients more access because access to trials is a big deal.
That's great. And then this kind of follows up with that with Alexandra asking, when realistically will combo therapies be available in trials, and how can overseas ALK patients work with their oncologists to potentially facilitate access?
Yeah, another great question. So I can't speak as well to how combinations or any drugs are approved outside of the United States because I'm not as familiar with the EMA or the Japanese FDA. So every country has a regulatory agency that is analogous to our FDA. I think really, in my mind, the biggest barrier is just making the trials more widely available, because not everyone can go to Boston, or not everyone can go to Nashville, not everyone can go to California. So how do we make it these trials? I think the combo trials would go faster if we could speed up enrollment, but that means having, you know, more broadly available study options. Outside of the United States, I can't speak as easily to how the regulatory officials work there. But usually once a drug is proven to be safe, or safe as a single drug or in combination, then other agencies will follow suit. For example, if something is approved in the US based on US clinical trial data, the same data can be evaluated in Europe or Japan or China. It's just a matter of different regulatory agencies that have oversight.
Good to know. Erica Lovacek asks, what research are you excited to see for ALK lung cancer coming up at the World Lung Conference?
Yes, so several things. So, you know, beyond, and I'm gonna really frame my question here around implementation science. And by that I mean is, it's fantastic to have great laboratory science, but if we can't actually deliver that great science to people, then what are we doing, right? And so some studies I think that will coming out of World Lung are really around real world, what they call real world data, is who's getting tested for ALK? How are they getting tested? What are the gaps? We want to be able to deliver effective therapies to everyone, right? So you can't, again, this is Christine editorializing, you cannot miss ALK. You just can't. If you're an oncologist, you can't miss ALK, because that opens completely different therapy options for your patients. But there are still some people and places that don't uniformly test. And so to me, you know, building data around what are people doing outside of clinical trials in the quote real world is incredibly important, because we want to be able to deliver what we have right now. And that will, in my mind, is probably the most impactful thing that will come out of World Lung. Yes, there's going to be beautiful science. There's going to be lots of combination studies, but until we can deliver on what we have, it's going to be hard to make headway on what's coming down the pipeline. And so I'm a huge believer in, we have to put lots of effort into making sure that we're getting testing, and that, this is importantly, that people are understanding the testing. Because there's two sides to that coin. We talk a lot about, is everyone getting tested? But the other part is, are people understanding the testing and delivering the therapies in the right context? So I get a lot of phone calls around, hey, Christine, I found this ALK mutation. There's no rearrangement, there's no fusion. You know, is this patient eligible for ALK? And that's a different scenario than talking about an ALK rearrangement. And so I think we really have to focus efforts on delivering on what we already have and get that to 100%. And so I'm excited to see a lot of these real world studies that are coming out that will show us, how are we doing? And
updates, yeah.
We can do better, and we need to do better.
That's great. There are a lot of questions about therapies not related to Western medicines, massages, chiropractors, acupuncture, organic supplements. What are your thoughts on all of this?
Yes, so I'm going to say this is something where I can't give you an evidence-based answer. So I can't say, oh, there's a clinical trial that said acupuncture was great for you. So I'm going to answer this a little bit from my personal perspective, which is that why not? If massage makes you feel good and acupuncture helps you, you know, I don't really see harm in it. And so I think when you start talking about chemicals or drugs that you're ingesting, that's a different scenario. And what I usually tell my patients is, if you're taking something that's over the counter, just let me know. Not judgmental, like I just want to make sure it's not going to interfere with the metabolism of your chemo drug, your pill medicine, whatever the patient is on. But I personally think things like massage and acupuncture and yoga really help our minds and our bodies. And so I don't see a downside to any of that.
Perfect. And then kind of a follow-up, Cherry asked, essential oils and diffusers. What are your thoughts? Are those okay, do you feel, for people to use?
Yeah, I don't have a great answer to this question, I'm going to be honest. You know, I think vapors in the air, you're going to inhale them. And so while some essential oils may have medicinal properties. I don't know what the effects of, how much are you inhaling over what period of time. I wish I had a good answer to this question. I don't. I think anything in moderation is probably okay, but I just can't answer in a data-based way.
Gotcha. JT asked, it seems that the biotech space is accelerating pipelines with CRISPR gene therapy and NK natural killer cell therapy, but not much from the academia space or on using these therapies in a clinical setting. What is your insight on this? And will the Lovely Lab be looking into this type of research? And can you also explain, because some people are not familiar with the CRISPR therapy?
Yep. So CRISPR was the Nobel Prize for 2020 to two investigators. Basically, what CRISPR is really amazing science. Actually, is getting rid of a gene. And so CRISPR is literally like cutting out a gene. And the CRISPR is the science and how you do that. And the reason it got a Nobel Prize is because it's really revolutionary. And it's actually something that viruses use that very smart scientists have said, oh, well, this is how nature does it in viruses. Let's see if we can sort of utilize that same system in humans. And I'm oversimplifying it, but it is beautifully simplistic in that all the CRISPR technology is getting rid of a gene, cutting it out in a very strategic way. We all use CRISPR. It's just part and parcel of laboratory work in 2021. Bringing it to the clinic is a little bit more difficult because how do you deliver it is the problem, and how do you make sure you get rid of the gene in areas that you want to get rid of it in, but not in other cells of the body. And so I think, there's been some rare diseases outside of cancer where CRISPR technologies have actually resulted in approved therapies. We don't have any cancer yet, but I'm sure it will get there. NK cells is another part of your immune system. So we think a lot about B cells, which make the antibodies, and T cells, which fight the infection or fight the tumor. NK cells are another way that we fight anything foreign in our body: bacteria, viruses, COVID, tumor. And NK cell technology is basically saying, let's like add another tool to our toolbox, and instead of just thinking about B cells and T cells, let's think about how we can use NK cells. There are early trials with NK cells. A lot of these like immune therapy trials, when you're talking about cellular therapy, like NK cells have been moved forward the most in liquid tumors. And for practical reasons is those tumors are floating in your blood. And it's easier to deliver a cellular therapy to the blood than it is to ask that therapy to get to a tumor in the lungs. I believe it will get there. I am not an expert on NK cell technology, so it's not something we do. We don't make NK cells in my lab, although Portia, my MD PhD student, is really thinking a lot about NK cells in the context of small cell lung cancer, which is very different than non-small cell lung cancer, as everyone knows. But I would say this is something that's very exciting, and stay tuned for more in the future. I think another really one that didn't come up but kind of related is something called bispecific T-cell engagers, or BiTE therapy, where you're basically trying to get a T-cell, a tumor fighting cell, or an infection fighting cell to a specific tumor target. So you imagine if you have a way to connect the T-cell to the tumor cell, that's what these bispecific therapies are trying to do. And those are moving forward in the cancer world as well.
Perfect. Ken is asking, do you feel federal lung cancer research funding is being fairly allocated, and should the pharma industry or the academics be the leader in this research?
I think we need everyone. And so we need the government, because that is predominantly how academic labs are run. We'll get back to funding in a second. We need big pharma because trials are overwhelmingly run by big pharma because of the cost and infrastructure needed to run a clinical trial. Is there enough federal money going to lung cancer? Well, I'm biased, but I would always say no. You're talking about the #1 cause of cancer-related deaths in the United States and worldwide. You can't put enough money or great minds behind this problem. So I think that right now I've really seen the lung cancer world pivot, and there's everything that's exciting about cancer right now, whether you're talking about targeted therapies or talking about immunotherapy or talking about early detection. It's all lung cancer. Everything is moving forward, and like everybody wants to be a lung cancer medical oncologist now, because all the cool stuff is in lung cancer right now, but it can't move forward fast enough. And it's certainly, federal funding is very difficult, and something we should talk about for a minute, which is how long does it take to get an NIH grant? If I write a grant today and submit it tomorrow's January 25th, when will I actually see those funds? If the grant is even funded. So the funding line is around 8% right now, meaning 8 out of 100 grants get funded. And if they get funded and I submit that grant tomorrow, I won't see the money for at least a year. That's a long time in the research world, where you're talking about, I need to get therapies better for patients now. I don't want to wait a year to really move this forward.
And that's another reason why, our ALK Positive group and our fundraising is so important. We started November 1st for Lung Cancer Awareness Month. And then, we raised 1.8 million, and hopefully we'll have that ready to go, and Colin can talk on that and by October, November. But we need more of it. Stian from Norway is asking question about epigenetics, and you've mentioned this on our labs several times. And maybe you should be the one to talk about what is epigenetics, but does all ALK positive cancer seem to have its own methylation identification? And is it possible to predict progression through looking at methylation in DNA from the circulating cells in blood? And can alternative pathway activations also be found through looking at methylation?
Yes. So, hi, thank you for chiming in Norway. It must be now the next day. So it's Sunday night where I am. It's probably Monday for you. So thank you for chiming in so early in the morning. Very exciting to have so many people here. Yes. So methylation, super cool. So we talked about, DNA becomes RNA, becomes protein, and there's layers and layers and layers and layers of regulation on each one of those. One way that DNA is regulated is something called methylation. It's basically like a tag on the DNA. So if you think about DNA as the instruction block for your cells, it's basically the map. The DNA is the map. It tells the cells what to do. The methylation is one way the map can be modified. And so, and let me give you a real world example. And this is one I like to use a lot. And one of my dear friends, Ricardo, I've used this example with him, and I saw him sign in. So if your tumor cell, imagine that you're trying to go to the grocery store and your normal pathway, your normal road is blocked because there's an accident. So how do you get to the grocery store? You need to get your milk, right? You're going to find a way around the accident. You're going to go an alternative route. There are lots of alternative routes that tumor cells can take, or cells in general, to get to do what they want to do, which is grow. And so one alternative route, if you will, is changing the DNA by putting this tag, this what's called methyl group on it, and just changes the way the DNA looks. And so that can change the whole cellular programming. It can totally change, instead of going down your direct route to get to the grocery store, you bypass the accident, you go another way. A methylation can change the same way. Like instead of going the normal route the cell goes, methylation can make the cell go another route. And so methylation is not just one methylation, it's usually hundreds or thousands of them. And yes, that is something we can detect in the DNA of a tumor or in circulating tumor DNA. And yes, I really am very excited about using methylation status in blood to help us detect response and resistance. I encourage everybody, next time you're at your physician's office, if you don't look at your scans, definitely look at them, because one of the biggest things I think we struggle with in cancer, especially lung cancer, is CT scans can be ambiguous, right? You look at a spot on a CT scan and you're like, is that a tumor? Or is it a scar from radiation? Is it a lymph node? Is it a pneumonia? I'm not really sure. That's not good enough. We need more granularity around, here's what I'm seeing on the scan. And one way we can do that, I think, is by matching the scan to changes in the blood. And so if a scan, well, maybe there's a tumor growth, maybe it's not. If the scan is ambiguous, but you see something in the blood, like a change in DNA level or a change in methylation status that's increasing, that may make you more likely to be like, oh yeah, this tumor is progressing. Because CT scans, look at them and talk to your physicians about them. They are ambiguous sometimes. more often than not, the more often than we would like. And so I would love to see a complementary marker be available for us. And I'm very, very excited to see ctDNA be one of those. And ctDNA is not just looking at mutations, but then also looking at methylation status in ctDNA. And ctDNA, for anyone who hasn't heard of it, is basically circulating pieces of tumor DNA in your blood that we can actually do a blood draw and detect the tumor DNA in your blood.
I remember one time Dr. Kamich mentioned during a longevity conference when he came, he said, if your oncologist is not looking at scans with you during your appointment, it's time to get a new oncologist. So yeah, I think it's important to do that. Those are most of the questions that people asked when we asked, but there's a lot of questions in the chat. Do we have time for a few more questions with you? Thanks. And another hot topic is the COVID vaccine. You know, I don't know, there's very few, if any, who have actually gotten the COVID vaccine as cancer patients. What are you hearing around your cancer center about how can we get it faster?
Yeah, so I'm going to answer that on two levels, which is what I see and what I know from like email and the lung cancer community. So what I see, and I had clinic on Friday, is huge variability in people who have gotten it versus not gotten it based on, you know, some of my patients are from Kentucky, some are from Tennessee, some are from Alabama, and the implementation across different parts and even in the same state is highly variable. And so this is clearly a huge issue to tackle. I will say basically every lung cancer organization, so I'm on the scientific advisory boards for longevity, GoTo, and Lung Cancer Research Foundation. The foundations have been working together to put statements with IASLC, the International Association for the Study of Lung Cancer, to basically, there's multiple letters that have gone out to multiple different agencies saying we need to prioritize lung cancer patients for the vaccine. We need lung cancer patients to be in a higher tier regardless of their age because of the risk inherent with COVID and lung cancer. And so actually you're going to see some movement in this week in the lung cancer field because you heard it here first. There is going to be a letter going out to the society that basically makes the recommendations. And our dear friend Amy, who is on the call right now, she's a big part of this, is making a joint statement across all the lung cancer advocacy organizations to tell the institution that basically puts out policies for vaccines or makes recommendations for implementation, is basically saying we need to prioritize lung cancer patients, make sure regardless of their age that they're in a higher tier because of the inherent risk. I would say right now, another question I get a lot, and this is a great question, when do I take the vaccine? And with respect to my therapy, should I take it, if I'm getting chemo, should I take it before chemo, after chemo? How do you time that? Right now, what we are telling, and this is again, not Christine Lovely speaking, this is Christine Lovely speaking as part of a large consortia of lung cancer doctors across the country saying, if you get offered it, wherever you are in your therapy, talk to your doctor and take it. Because we don't have any data. Is it better to get it before chemo or after chemo or during? I mean, we don't have that data. And the most important thing right now is just getting the vaccine.
That's great. Let's try to get a couple more questions. I keep hearing, Gina, is there any potential for virtual trials with combination therapies? Could we jump on the technology that we have gained from telemedicine and COVID-19?
For sure, yes. And I think actually one thing I wanted to talk to you guys about as we end too is a way that people are doing that. I think there's going to be more and more and more virtual consenting and what we call single patient IRB. So IRB is Institutional Review Board. That's the group of people who basically review a trial and say, okay, we think this is safe or not safe. There's a lot of paperwork that goes into implementing trials. But if we can say, let's say, for example, there was an ALK positive patient in Wyoming who couldn't make it to a big academic medical center, you know, how do we get that patient on the latest, greatest therapy? Can we do that? What does that mean? How do we implement that? Well, first is what we call this single patient IRB, where you have approval to give somebody an experimental therapy. Second is all the consenting, which probably everybody on this call has done e-consenting at this point. So that's an easier box to check. And then it's delivery and oversight of the drug. And there are companies being formed that have nurses that can go out to the public and be like, hey, I'm going to go monitor this patient for toxicity on these drugs or side effects on these drugs. And then you report back to a doctor in some sort of infrastructure like that. So I'm super excited to see this move forward. I'm a big believer that we need to think outside the box on how we deliver excellent care. It's ridiculous, and sorry, I'm being really passionate about it, but it's ridiculous to tell people that they have to pick up their lives and go across country because they want to get the best cancer treatment that's available for their type of cancer. That's absurd. It's not practical. It's not sustainable. It's not affordable. And we can do better, and we have to do better. And I think one sunlight that will come out of COVID, one silver lining will be everything that we had to completely frame shift for COVID in medical care will carry over to other aspects of medical care after COVID. We already do telemedicine. Half of my visits on Friday in clinic were telemedicine and half of them were in person, and that's awesome. And so, yes, I think it will roll out. Over what time period? I'm not exactly sure. I can tell you I'm actually involved. So Roche is running one of these trials for ALK, and they have to implement this kind of trial since it's so novel with drugs that we know a lot about. And so Roche Genentech make ALKtALK. There, I'm leading a study with them that is exactly this kind of trial. It's working with Foundation Medicine and Genentech and ALKtALK. And so basically, if a patient with an ALK positive tumor that's not lung, so let's say a patient in Wyoming has ALK positive breast cancer, can we get them an ALK inhibitor on a clinical trial, a one-off clinical trial, so we can learn about using these drugs in other types of ALK positive lung cancers? And I think it's all with remote consenting and nurses that actually go out to the patient's home to monitor them, virtual formats. And I think two things about this trial: it's totally novel in how it delivers care, and it's also bringing a drug to other patients. So yes, we know about ALK positive in lung cancer, but only this month did an ALK inhibitor become FDA approved for ALK positive another tumor type, which in this case was a lymphoma. And so why did it take so long? If we knew about ALK for just as long in lymphoma as we did in lung cancer, it was because there wasn't enough patient data. And so how do we get enough patient data to actually deliver on drugs that we have right now for other indications? So in this case, it's if a patient has ALK positive colon cancer, I can't write them an ALK TKI in clinic because the drug is not FDA approved for ALK positive colon cancer. But is it the right therapy to do? Well, we won't know until we actually start treating patients. And so that was a really long answer to your question, and I apologize, but the take-home message is yes, absolutely. Paradigms that came to us because of COVID will cross over post-COVID.
That's great. Well, I want to make sure we're respectful of your time. So I think on behalf of all of us, thank you so much. And you have to talk about Mr. T.
Here's Mr. T. This is my dog Tegan. He's like my #1 family member. And everybody who knows me knows Tegan, so he wants to say hi.
And I think it's so interesting. Tegan is also on a targeted therapy pill.
It is. Tegan he has a tumor called mast cell tumor, and he takes a targeted therapy called imatinib. So yes, this dog mom believes in TKI so strongly that she takes the dog with mom.
That's great. So everybody unmute yourself. Tell Dr. Lovely, thank you. We are so appreciative for all that you do.
Thank you.
Thank you, Dr. Lovely.
Thank you very much.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you so
Thank
much.
you.
Thank you.
Thank you, Dr. Lovely.
If everyone wants to stay on, feel free. We do have this until about 8:30. So, but Dr. Lovely, thank you so very much.
Thank you all so much for being here on your Sunday night.
Thank you.
Bye.
Thank you.
Thank
Appreciate it so much.
Oh, she is so awesome, isn't she? She has such passion for our ALK community and has just been so amazing to open her lab to us several times. I just love her to death.
It's amazing.
I do too. And that combo therapy, I don't know, gives me hope, that we're moving in the right
I'll
direction.
come down later and turn the water on.
And already seeing like some results with clustering. I thought that was really impressive. Is there science people on here who wants to talk about clustering or